those of you who join us today at the Governor Hotel, and those of you listening on OPB Radio or watching on Portland Community Media. Today we'll hear from Charles Wilhoite, Chair of the Oregon Health and Science University Board of Directors, and Dr. Lewis Picker, Associate Director of OHSU's Vaccine and Gene Therapy Institute. But first, some announcements. The generous support of City Club's corporate and media partners ensures we put on the state's best civic programs week after week. I'd like to thank our media partners, including Oregon Business Magazine. Our current Friday Forum sponsors are Geffen Mesher, Iberdrola Renewables, Perkins Coie, Portland General Electric, and Providence Health and Services. We are grateful for your support and commitment to City Club's mission. Please join me in a warm round of applause for all of them. Next week, we'll broadcast live on OPB's Think Out Loud from the Governor Hotel to discuss the results of the Oregon Values and Beliefs Survey, Oregon's most recent effort to speak with all Oregonians about what they really think, really value, and really believe. Doors will open at 11:15 and the program will begin at 11:50. You can learn more about this and other club events, purchase tickets, and become a member on our website, pdxcityclub.org. As always, we'll be live tweeting today's event. You can follow or mention us at PDX City Club, and be sure to use the hashtag CityClub in your own tweets. We'll be having a Q&A session with Dr. Picker at the end of today's program. Members, please come to the microphone to ask your questions. And for all our audience participants, please locate the index cards on the center of your tables and write your questions on them during the forum. We'll collect them prior to the start of the Q&A. And now to our program. As the state's only health and research university, Oregon Health and Science University brings together patient care, research, education of the next generation of healthcare providers and scientists, and community service to improve the health and well-being of all Oregonians. Our featured speaker today will be Dr. Lewis Picker, Associate Director of OHSU's Vaccine and Gene Therapy Institute. But first, to set the stage, we'll hear from Charles Wilhoite, Chair of OHSU Board of Directors. Mr. Wilhoite started serving on the board in 2004 and has chaired since 2010. He was appointed to the position by Governor Kulongoski. In addition to his service at OHSU, Mr. Wilhoite is Managing Director at Willamette Management Associates Portland office and National Director of Tax-Exempt Entity and Healthcare Services. Please join me in welcoming Mr. Wilhoite to the stage.
Thank you very much, John. Good afternoon. Once again, my name is Charles Wilhoite, and for the last several years, I have had the privilege and the honor to serve as the board chair for Oregon Health and Science University's Board of Directors. As John stated, I was appointed by Governor Kulongoski in 2004, and upon being appointed, I quickly realized that OHSU represents quite possibly one of the most complex organizations you could ever encounter. And I say this after having spent the last 30 years of my professional career doing nothing but analyzing companies. So it is a very complex organization run by a high level of qualified professionals who basically make my job as chair very easy. And that might surprise you, but I would have to say it's nothing but a pleasure to serve as the board chair at OHSU. Rachel asked me what my primary role is as the chair at OHSU. And I laughed to myself because I realized that my primary role probably is keeping myself and the other board members out of the way of all of those smart people up on the Hill and throughout our system so that they can keep doing all of the marvelous things that they do. Today, thanks to the invitation from Sam Adams and the crew here at the City Club, I have a few minutes to give you a broad overview of OHSU before we get to an actual scientist and today's headliner, Dr. Picker. I will touch briefly on 4 topics vital to the future of OHSU and to the health of all Oregonians. They are philanthropy, partnership, diversity, and leadership in the transformation of healthcare. I put philanthropy first on the list because of the incredible news and the incredible challenge articulated just last week by Phil and Penny Knight. Simply stated, the Knights made it clear to us that if we could raise $500 million for cancer research in the next 2 years, they would match it. And that is an amazing pledge. I would even clap at that again. And because I take my board responsibilities, and particularly my chair responsibilities, so seriously, I'm going to take a moment here to ask if there's anyone in the audience who has $500 million that they might give to me. It's important to me as a chair to make this a reality before I step off the board, and I know we're in Oregon and people are pretty modest in Oregon, so I'm going to be here a few moments afterward. I know no one wants to get all that attention, so come up to me and let me know, because that's a critical goal of ours. When we look at OHSU, clearly we're thinking about the future all the time. And we share a common goal with the Knights. We want to end cancer as we know it. And we've already put some dents in cancer's armor. Dr. Brian Juker, head of the Knight Cancer Institute, and his team worked to create the first drug, Gleevec, that stops certain types of leukemia without the side effects of chemotherapy. Dr. Drucker and his team are pioneering new efforts in what we call personalized medicine, and that is pioneering medicine based on your own gene sequence and the genetics of your particular cancer to get us closer to that day when we can customize your treatment in the most effective and the least damaging way. None of this, of course, would have been possible without philanthropy, without the Knights' original gift of $100 million, without the research support of the Leukemia Lymphoma Society, or the smaller gifts of thousands of individuals who helped OHSU reach a record in philanthropy last year of $197 million. Our work in cardiology, health and nutrition, dentistry, and pancreatic cancer, neurology, medical education, all of it is made possible by philanthropic gifts from leaders in our community, such as Ken and Joan Austin, Bob and Charlie Moore, Bonnie and Eugene Skirties, Howard Volum, and Arlene Snitzer, and others. So first and foremost, OHSU's growth and success in the last decade has been spurred by the giving of these community pillars and by the consistent support of thousands of others. We are proud of these partners and their support, and they are critical to our continued success. As we rely on philanthropic partners, we also rely on academic and institutional partners and collaborations. I will mention one giant example of that rising on the banks of the Willamette River between the Ross Island and Markham Bridges. Called the Collaborative Life Sciences Building, it is a $295 million joint project between OHSU, PSU, and OSU. And I think it's safe to say that there's not another academic project like this in the country sponsored by 3 institutions. So we're very proud of our willingness and our history of collaborating with others in the community and throughout the state. When it is finished, this building will represent 600,000 square feet of labs, teaching space, research space, and the home of our entire School of Dentistry. And we hope it will become a place that is viewed as only a community asset, a place to learn and share, and a place that anchors our expansion into what we call the Snitzer campus, based on the Snitzer family's donation of the land on which OHSU hopes to expand in the coming decades. OHSU is also embracing change occurring all around it in our larger community and in the transformation of healthcare. Central to our strategic plan is the concept that OHSU must be an institution that is diverse in ideas and people if it is to succeed. Speaking from personal experience, I will say that can be a challenge in Oregon, but it is a challenge that we are meeting head on. The population we serve is changing rapidly. The talent pool from which we draw is not local, not regional, not national, but global. And the collaborators and academic partners, and though I just mentioned 2 locally, are just as likely to be in Asia and Europe as they are to be in Oregon. OHSU has put this cultural imperative, which is to embrace the power of difference, at the center of its plans, and I am confident that we will succeed in creating an institution as diverse as the world we serve with our education, healing, and discovery. Finally, OHSU is helping lead locally and nationally in the transformation of our healthcare system and to deliver better outcomes for more people at a lower cost. Locally, we are a key partner in our region's coordinated care organization, HealthShare Oregon, and our president, Dr. Joseph Robertson, plays a key role on Oregon's Health Policy Board. Nationally, however, our teachers and scientists play a lead role in figuring out what works in healthcare transformation. Our Center for Health Systems Effectiveness is a leader in assessing how well innovations in our state and others deliver results. We have one of 8 centers in the United States that studies medical best practices. How many tests are too many? When do you really need an MRI? These questions may seem basic, but in the aggregate, they can change the actual practice of medicine to the benefit of the patient, both in costs and outcomes. So once again, I am very proud to be able to be a part of this great institution that just celebrated its 125th anniversary. We are large and sometimes feel far-flung, but we are focused on a mission that is simple: improving human health. With your support, we will continue on that mission, producing results like those you will soon hear about from Dr. Picker. Thank you all for helping make OHSU the great institution that it has become. With that, I would like to introduce you to Dr. Lewis Picker. Dr. Picker serves as the Associate Director of OHSU's Vaccine and Gene Therapy Institute. He is a graduate of UCLA and received his medical degree from UCSF. He completed his residency training in pathology at Beth Israel Hospital in Boston and postdoctoral training in immunology at Stanford University Medical Center. After a stint at the University of Texas Southwestern Medical Center in Dallas as an assistant professor and then an associate professor, Dr. Picker joined OHSU in 1999 as professor of pathology, molecular microbiology, and immunology in the OHSU School of Medicine, and head of the Division of Pathobiology and Immunology. See, that's what you really have to learn as a chair. You've got to learn to say all these words. As you are probably aware, Dr. Picker and his team have been making significant strides towards developing a vaccine to rid the body of HIV. Needless to say, Dr. Picker is a very valuable member of the OHSU team. Please join me in welcoming to the stage Dr. Lewis Picker.
A little slowed down by a hip fracture due to typical Oregonian activities, mountain biking. But the good news is I only have one leg to stand on, so I can only talk half as long. Okay. I have a few, let's see, a few slides here. It's not showing up here, maybe. It's not my computer.
It's not the computer. Well, it would be a fascinating presentation. Is the AV leader here?
Okay, well, we could start, I guess we could start without it. It's the slides are hooked up on the computer, but they're not hooked up to the screen. First of all, I'd like to thank Charles for that nice introduction. And it's really an honor to be here at the City Club. I've often listened to the broadcasts on OPB and never in a million years imagined that I would be standing up here. But in a sort of a discussion that's usually focused on public policy. But in a way, what I do is also an issue of public policy, because if it wasn't for vaccines that we all take for granted, we would be clamoring for our elected politicians to do something about these infectious diseases. There are 50 or 60 or more licensed vaccines. Here we go. As shown in this slide, to which we depend on. It's estimated that these vaccines that save about 3 million lives a year and countless millions more in people who would have lifelong disabilities due to these terrible diseases. In red arrows, you notice some of the greatest killers of mankind, such as smallpox, which is not something that we worry about anymore in 2013. Typhoid fever, poliovirus, and iron lungs. Medical science has eliminated these diseases from our common lives, and probably the the production of vaccines is probably medical science's greatest triumph in terms of human health. But sadly, HIV, the scourge of the last century and still of this one, is not on this list. There are currently 34 million people with HIV and about 1 million deaths due to AIDS every year still, even in this era where we have drugs that can treat the viral infection. So the 2.5 million new infections that occur every year could be prevented if there was a vaccine, and we would almost double the lives saved if we could develop an HIV vaccine. Now, in this talk, I'm going to give you a little bit of history and a little bit of science. Don't worry, there won't be a test at the end. But let's start with the history. HIV was actually discovered as a virus in 1983. The disease AIDS was first described in 1981. I was a senior medical student at the time. And with the discovery, there was an announcement by Margaret Heckler, the then Secretary of Health and Human Services, that within 2 years, an AIDS vaccine would be ready for human testing. And of course, this was at the beginning of the molecular biology revolution, and it was thought that, hey, we can identify a virus, we can make a vaccine to it. And this promise has sort of haunted the field ever since. So it's been almost 30 years, or it's actually been over 30 years now since the isolation of HIV. There is still no effective vaccine. And so in this talk, what I'm going to try to explain to you is why there is no vaccine, and what are the chances that a vaccine will develop. And are we really as off schedule as we would think based upon that 2-year promise? And as often the case, the past can offer insight into the future. And I'm going to talk a little bit about smallpox, because really smallpox is an epidemic scourge which serves as a model for what we're dealing with HIV. This is a very old disease. They found evidence of it in Egyptian mummies. It was thought to cause the plague of Antonin in 108 AD, which was thought to be the beginning of the end for the Roman Empire due to the millions of deaths that this disease caused. In 17th century Europe, which seems like a long time ago, 400,000 people died annually from this disease, and 1/3 of survivors went blind. 20 to 60% who contacted died. Now, it's been known since before Christ that survivors were immune. If you survived the disease, you could then care for the sick. And as far back as the Middle Ages, particularly in China, people were experimenting with controlled inoculations of actual smallpox material from sores in order to mimic that protection that you get when you survive. This process was risky. About 2 to 3% of people actually died from this initial treatment. But nevertheless, it was still a benefit to have 2 to 3% die than the 20 to 60% die if you actually get the disease. Now, this all changed in about the late 1700s, 1796, when Edward Jenner, an English physician, had been hearing stories of that dairy maids were immune to the disease. And actually had been hearing stories of people actually taking lesions from dairy maids and inoculating their children with it in order to protect them. So in May of 1796, Dr. Jenner found a young dairy maid who had a disease called cowpox, and took a lesional material from this individual and gave it to a young boy named James Phelps, or James Phipps. The young boy got sick for a little while and then recovered. And then later on, there was no FDA in those days, there was no regulatory structure, Edward Jenner just simply decided, well, he was going to inoculate this young boy with actual smallpox material. And lo and behold, he was protected. He did not get sick at all. So again, without an FDA to regulate him, Dr. Jenner went on and did this to 23 others and conclusively proved that transfer of this material from the cowpox to protected from smallpox. And this discovery was really what ushered in the modern era of vaccination. What Jenner didn't realize he was doing, because he didn't understand where these diseases coming from, that this was actually a virus, a cow virus, that was very closely related to smallpox, but because it was adapted to cows, did not cause disease in humans. And he called this treatment vaccination after vacca, which is Latin for cow. So that's how this whole vaccination era started. And it really changed the course of history. This new technique was much safer than the old technique of that 2 to 3% of people died. In fact, 1 in a million have problems with this vaccinia vaccination. And it really changed the practice of medicine, ultimately leading to the complete eradication of smallpox in the 1970s. And it ushered in an era of vaccination against deadly infectious diseases. And here's just some of that history. And it's interesting to note that despite the fact that Jenner had clearly demonstrated the value of giving a weaker form of a pathogen to protect you from the stronger form, is it took decades for the vaccines that we've come to depend on to be developed. Louis Pasteur in the 1880s, that's almost 100 years later. Max Tyler in 1936, yellow fever vaccine. Famously, the 1950s, the Salk and Sabin vaccine for polio. And then finally, Maurice Hillerman, who worked at Merck, developed many of the vaccines, greater than 40 at all, that we depend on today as our childhood vaccines: measles, mumps, rubella, chickenpox, and so forth. And these are the vaccines for which we rely on, but it took years and years and years to figure out how to make them. And this impressive menu, in many ways, was really the low-hanging fruit, because all of these diseases, all of the ones for which the vaccines have been made, it was clear that people who recovered were immune. People who recovered were protected. So that means the immune system, it was known, had the capacity by one of its various mechanisms to combat that pathogen. And there are two mechanisms that the immune system uses. One is antibodies, which bind to the pathogen and activate it, and the other is a cellular immunity mediated by T cells, which recognizes infected cells and kills them. So in simple fashion, that's how the immune system deals with viruses. But in all these cases, it was clear that the immune system had mechanisms to deal with these viruses or bacteria. But this is not true for all viruses, because we have been unable to make vaccines with agents that cause chronic diseases, which have evolved to evade the immune system. And the AIDS-causing viruses are the poster children for chronic persistent agents. The AIDS virus, HIV, originated as SIV in African monkeys. This virus evolved in African monkeys to infect the monkeys for life. The monkeys, in fact, had to adapt their entire systems so that they didn't get sick from these viruses, but they were never able to get rid of it. When the viruses are transmitted to humans, the mismatch is such that it actually causes AIDS. So instead of it retained its ability to persist in humans, and in fact, infections heretofore have been thought to be completely permanent, but because of the mismatch, the fact that we're not adapted to the virus, over years, our immune system gets degraded and we get AIDS. And it's the same is true for Asian monkeys, the rhesus macaques, which form the experimental model for HIV disease. So the ability of these SIVs to completely evade the host monkey's immune system and the ability for HIV to evade our immune system has really been a problem in terms of vaccine development. There is just no protected state. Everybody who gets HIV always has the virus, with a few very, very interesting exceptions, bone marrow transplant and so forth. But for the most part, there is no natural immunity to these viruses that can eliminate the pathogen. And that's in great distinction to other viruses that we've talked about. Now, in 30 years, the field has taken the time to figure out why this is, and it turns out that HIV really has multiple weapons. First of all, it kills cells that the immune system depends upon to mount a response, so-called CD4 T cells. Secondly, it hides silently in our cells, so it has a latent phase where it can sit there and be unnoticed by the immune system and then come out at later time points. Third, it's actually evolved its important bits on its surface, the proteins that it uses to enter our cells. It hides the important parts underneath a camouflage so that they can't be reached by antibodies. And finally, it has an impressive ability to escape. So if you do have an immune response that works, the virus will almost always evolve around it, change itself because it's so malleable, and escape. So this is a really difficult target. The other thing we have to deal with is that if you look in the left, those little squiggles on the left, that's the variability that we see in the global flu viruses. And if you look in one patient, there's probably 10 or 20-fold more variability of HIV viruses in one patient than there is in the entire worldwide array of flu viruses. And we have to get vaccines against flu every year because of the variability. And of course, if you look at all of the HIV types in the world, the variability is tremendous. And a vaccine has to be able to recognize all those types. And finally, to make matters worse, is this virus is capable of replicating extraordinarily rapidly. On the upper right corner, you see there's some tissues with all the brown spots are virally infected cells. That is 10 days after the infection. There's an extraordinary invasion, extraordinary level of infection, and that is before the immune system can even get to it. So the immune system is trying to parachute into a battlefield which already has been completely taken over by the enemy, because the immune system takes several weeks for it to mature and develop sufficient antiviral effective activity to work. It's too late, too little, too late for HIV. So the field has been busy. It's not like the effort to try and make an AIDS vaccine, that we've all been sitting and twiddling our thumbs. We've tried all the usual ways that people make vaccines, all the way that Pasteur, the Maurice Hellerman, that all of the Salk and Sabin, we've tried all those tried and true ways. We've tried inactivated viruses. We've tried attenuated viruses. We've tried protein subunit. We've tried other vaccine vectors that are so-called prime boost. We've tried combinations of the above, and they've all been either ineffective, too dangerous, or not sufficiently effective to make a dent in the clinical problem. All of these have failed. So I came into this, I started working in HIV probably in the mid-90s. I've always been an immunologist, but I switched my lab's work to focus on HIV in about 1995. Was thinking hard on the subject and decided about 12 years ago that we might have a plan that could do something that could be a little different than what the field had done in the past. And in fact, to implement this idea is why I moved to OHSU in 1999, is because of the fact that OHSU has a primate center to test these ideas and had other experts here for which I could work. So we hypothesized that the best way to deal with HIV is to hit it hard and hit it early, to have immune responses that are there all the time, so when the virus invaded the body, there wouldn't be a delay before the immune responses were there. The immune responses would be there on the beaches, so to speak, to man the beaches and not let the virus get a head start. Sort of a whack-a-mole, a whack-a-virus type approach. So we had to figure out a way to get T-cell responses or other immune responses that were going to be there all the time. And that's really the difference in the approach that we've taken here at OHSU. We looked for help from what I call a veteran gunfighter. We looked for a virus that would generate to itself the kind of responses we think would be useful against HIV. We wanted a virus that could generate armed T cells that would not be eliminated, that would persist and would keep these immune responses there all the time. And then, of course, this virus also had to be engineered so that it can express HIV antigens, so that it can serve as a vaccine vector to acquaint the immune system with HIV, so that those T cells could be, those immune cells could be able to recognize HIV when it came in. And only one virus met this criteria, and that's a virus called cytomegalovirus. Now, this is a virus that probably half the people in this room are infected with. Don't worry about it. You're not going to get sick. I mean, probably 99% of the developing world is infected with it. This is a virus that our bodies have learned to live with, and only rarely causes disease in situations of if you get it during pregnancy or if you get a bone marrow transplant. For the most part, it's completely benign. But it has a very unique relationship with the immune system that we noted and we brought with us in terms of our HIV vaccine development. And really, it can be considered a Goldilocks virus. It sticks in our body just enough, just the right amount. For its own reasons, and this is a virus that was present in mammals when we were being trod on by dinosaurs. So it's developed with us. We've co-evolved with it. But for its own reasons, it's developed, it likes to have lots of immune cells recognizing it. It doesn't want to kill us. So it elicits, on purpose, very high immune responses and keeps them armed and ready to go all the time. And presumably that's to keep the virus in check. It still does its thing. It still gets passed from person to person, but it's a very, very low level. And this low level is just the right amount to keep the immune system active, just the right amount. It's not too much to exhaust the immune system, and it's not so little that the immune system, like most vaccines, goes back to a resting state. Because that's the problem with conventional vaccine approaches. You get a bolus of exposure, and then your immune system goes back to a resting state. So CMV is, we thought, would be just the right level. And so we went ahead and to make a 12-year story short, we learned how to engineer this virus so that we could insert within it HIV, or in this case SIV proteins, because SIV is the virus we use in our model system. And we showed that in fact we could generate very high responses that were armed and dangerous, that had the phenotype and function of an armed T cell, such that they could man the beaches. If you notice, there's a graph here with all the level of responses and all the various sites of the body where HIV could enter, and they're all very high. Between 4 and 5 to 20% of the T cells are able to recognize SIV in vaccinated animals. So it turns out we were right. It took to test this hypothesis from beginning to end, probably took about 8 years. But now we've tested it exhaustively in non-human primates in the SIV model with a virus that's probably 50 to 100-fold more pathogenic than HIV is in humans. And we've shown that about 50% of the animals, as shown in that graph there on the left, group A, 9 out of 16, instead of getting massive viral replication upon exposure to SIV, the viral replication is strongly contained, and all you see is a few blips and then complete control. And even the one animal that relapsed with what's called elite control, what's conventionally called elite control. So those 9 of 16 monkeys were unprecedentedly protected against this very aggressive virus, which is often called the Death Star because of its pathogenicity. Of course, on the right, you see groups B and C. Those are the controls. They had no protection. And this effect was first reported in 2009 and again confirmed in 2018. What we reported very recently, and then just a few weeks ago, was really the outcome. So remember, I told you this is a virus that in 50 million human cases that over the years, and in all the cases in all the monkeys, it's a virus that has never been eliminated by the immune system. So it's 50 million virus, the score 0 for the immune system. And what we found in these, as we followed these animals that were protected for longer, is you notice in that graph over there, there's occasional blips. That's measurement of virus, and those go away. About 70 weeks, we no longer see those. And if you follow the animals out for 3 years, even with ultra-sensitive tests, you do not find the virus. And in fact, when you look in tissues of those monkeys, with the most sensitive test that medical science has, you don't find the virus, and you cannot distinguish an animal that was infected and then protected by this vaccine and an animal that had never been infected. And it's very different from an animal that has been infected and then completely suppressed by antiretroviral therapy. So the drugs that HIV-positive people take, the same drugs work in monkey. They suppress the viral replication, but they do not get rid of the virus, as you can see in the upper right-hand corner. So this is the first immune elimination of an AIDS-causing virus. And that's what it says there. So that's why the world has been sort of interested in this, and we've received so much attention in the past 2 weeks in the press. So, of course, this is all in a model system. It's a very high bar model system. It's very aggressive SIV, but the real proof of principle will have to come in humans. And as my initial part of the talk indicated, moving into humans is not quick. There are obviously safety concerns that we have to deal with. We have designed a virus, a human virus, that's suitable for a human vector that we believe has all the characteristics, the safety characteristics that will allow us, the regulators, to allow us to put it in humans. We believe it will have the same effect, and we believe it will work. We're engaging in conversations now to get this manufactured and to begin clinical trials. Of course, it's going to take several years to do so. So we're hoping for clinical trials starting in about 2016. And as that picture of the road shows, the road to a licensed vaccine is very long. Remember, that clinical trial is just the first trial in humans, and it's mostly a safety trial. It's still going to take a number of years after that to get it through efficacy trials and ultimately to get a licensed vaccine. But you have to start somewhere, and with OHSU's help and the help of all of our funders and the support of everybody in this room, I think we can get there. And with that, I didn't do this work myself, even when I'm not on crutches. There's no way I could do all these experiments myself. There's a large crew of people at OHSU who work on this project. In particular, in red is Scott Hansen, who's sitting in the audience here, who's my battalion, right there, my battalion commander, who runs the operation on the ground, and a number of collaborators and all of the various people who work on this project. I also have to make sure I acknowledge the funders, in particular the NIH and the Gates Foundation, who've supported a lot of the recent studies, because this is a very expensive research, and it couldn't be done without their support. And finally, I'd like to thank my family. Sitting in the audience is my wife, Belinda, 2 of my 6 children, Mikey and Zinzo, and without their support and allowing me the time to think about this occasionally, when I don't have to do chores, I wouldn't be able to be here. And with that, I'll stop and be happy to take any questions.
Thank you, Dr. Picker. And now, if you have a written question on the index card, please hold it up high so that City Club staff can collect it from you. We'll now take questions from the floor. As always, we invite members to the microphone to ask their question. Asking questions at the Friday Forum microphone is a benefit of City Club membership, and membership is open to everyone. Before asking your question, please identify yourself as a City Club member. Please ask only one question, and ask your question as succinctly as possible. If I flash the index card question mark, it means please wrap up your question.
Tom Hamilton, City Club member. Dr. Picker, what hypotheses do you have for why it's only 50% effective? What's going on with the other half of the monkeys?
That's obviously an area where we're intensely interested in. It may well be that we're dealing, as I mentioned before, it's up until now it's been 50 million to 0, and the fact that we're now 50-50 is actually a big difference. There are several possibilities. One is that the virus is just too pathogenic, that there's a battle that goes on at the initial port of entry, and sometimes the host wins and sometimes the immune system wins. Again, with this very pathogenic virus. It may well be that in humans it's 100% infected. We won't know until we get there. We don't see obvious differences between the animals that are not protected and the animals are. They tend to be a little bit better, the ones that are protected, in terms of the level of immune response, but it's not enough to be convincing. It could also be related to the genetics of the monkey, that because this is such an aggressive virus, that the monkeys that wear the virus just slow down a tad might be the ones that are more susceptible to infection. That's an area of active research, but in terms of translation, what we've done so far has really been a proof of principle that it can occur. And we're not going to know the details of how it's going to work in humans until we actually move to humans.
Richard Barker, City Club member. Dr. Picker, how have your initial results compared to some of the other research avenues in this area of study?
Well, there's obviously a lot of ideas out there for HIV vaccine development. For a while back, general conventional T-cell vaccines were all the rage. That just a so-called prime boost approach that has the disadvantage of making the T cells all go back to a resting state. That has been shown both in monkeys and humans not to work. So in terms of a cellular immune response, it's really our persistent vector with its effector arm T cells is really heads and shoulders above all other sort of T cell based vaccines. Now there are a number of other approaches to try and make antibodies that counteract, to get by the virus's defense against antibodies. The so-called broadly neutralizing antibodies that some patients who have HIV make. The problem is that we don't know how to make them yet by a vaccine. But that's also an area that a lot of investigators are working on, is trying to figure out how to make broadly neutralizing antibodies. And so ultimately, I think that these approaches are going to be complementary. This is a very difficult virus. As I said, it's evolved over tens of thousands, even hundreds of thousands of years to evade the immune system. We're going to need to identify all its vulnerabilities and probably hit them together in order to make a completely effective vaccine. So I ultimately see our approach maybe combining with a more successful antibody approach ultimately in the licensed vaccine. The one thing that's different, though, that our approach also has potentially the ability to be given as a therapeutic vaccine to individuals who are already HIV positive, who are treated on antiretroviral therapy. Because of this ability to clear the virus, we show there's definite clearance of the virus in these animals. This also might be an approach that would help the cure agenda, which is also very big in the field now, to try and actually cure people who have the disease, have the infection, so that they can go off drugs.
Doctor, could you speak on the economics of the future, both how they might benefit OHSU as well as the privately held company that's holding some of the patents, and is there going to be an opportunity in the future for the public to be part of the investment into this wonderful discoveries?
Well, obviously we try to get patent protection for our technologies. We move forward and to keep that IP safe so that when the time comes, a big pharma company will be interested in producing this and moving this vaccine out to the public. That's a ways away. I have a feeling that in order to get their interest, we're going to have some successful phase 1 trials, and at that point, I think that we'll see some big pharma partners appear to move this vaccine platform forward. And it's not only for, this vaccine platform is not just for AIDS, by the way. I mean, we're also investigating its efficacy against tuberculosis, for which there's not a very effective vaccine, and for malaria, for which there's no vaccine. For similar reasons of getting there first with the mostest, it may well be effective in those diseases. So this is a vaccine platform that will ultimately benefit OHSU by virtue of perhaps royalties, but at this point we're just focused on seeing if it works, getting it out there, and trying to help as many people as we can.
A related question from the floor is, what kind of funding is needed to move forward? And what else could you do if you receive more funding from sources such as philanthropy?
Oh, that's a great question. I wish Phil and Penny Knight would give us a challenge on that one. I mean, the part of the delay that are built into our projections of how quick this is going to go is that at every step I have to raise money. That's why I rely on Scott to do the work in the lab, because like a university president, my goal is to write, my job is to write the grants and to get the money. We have been in this preclinical phase, very generously supported by the NIH and the Gates Foundation, and we hope that they will continue to support us. AIDS vaccine is one area where you can get public monies to take it through to later, phase 1 and later phase human trials. On the other hand, if we had $25 million for which to move this forward, it would make it a lot quicker. So if we had philanthropy, if we had the funds to be able to pay the people, to pay the contract manufacturing organizations to manufacture our virus, to be able to pay the people who do the clinical trials, if we had that all lined up now, it would be a lot quicker.
City Club member, what is your actual timeline and what are next steps, and can you say any more about what you just said about the effect that money will have on that?
I didn't catch the last bit about the money.
What effect are the dollars actually going to have in the availability of funding in terms of the actual
I'll tell you
timeline?
what I'm doing, what I'm actually working on this week is grant proposals for our next phase. And the next phase consists of largely of taking our candidate vectors, getting them made under GMP conditions, to take them to the regulators and plan for a phase 1 clinical trial. We also have a number of questions we're trying to work out that we're still using the non-human primate models for, trying to improve the vaccine so that the candidates we bring forward are likely to be the most effective in human. And this costs a lot of money. I mean, the next phase is probably going to be, for the next 3 years, is probably going to be $10 or $15 million, maybe even $20 million to get through that first clinical trial. And then after the first clinical trial, assuming it is successful, we probably would have a second generation vector to test, and then finally efficacy trials. And that's when it gets really expensive, because that's when you have to start doing thousands of people in areas in which HIV is endemic in order to get an efficacy signal in human. Some of the more recent trials have had as many as 18,000 subjects or 16,000 subjects. We could probably do it in certain areas such as sub-Saharan Africa with fewer subjects, but nevertheless we're talking $50 to $100 million or more for that licensure trial. But we need to get there first, and we need to get there. We need to get into humans. We need to have a first-in-human trial that shows that the kind of responses that we see in the monkeys can be recapitulated by our human vector, the human equivalent vector, and that it's safe. And then that's what we're focused on right now.
Another question from the floor here. How should people interpret Magic Johnson's apparent ability to appear totally unaffected by HIV for so long? Is he a case study?
Well, you know, despite the promise of a vaccine in 2 years by Margaret Heckler, the field actually took a little bit longer, but by the late 1990s, the biochemists and pharmacological chemists had come up with antiretroviral therapy. These are drugs that can prevent the virus from replicating, and therefore, when the virus doesn't replicate, it doesn't cause the destruction of the immune system, which leads to AIDS. But those individuals who have to take those drugs every day because the virus just sits there, very high levels of the virus are sitting there in its cells and periodically replicating, and as soon as those drugs are producing more virus, and as soon as those drugs are taken away, the flame is always there, and that fire is relit, and they start progressing again. So, it's very magic that, you know, that Magic and others with HIV can have those drugs, and therefore we don't see AIDS like we used to in the 80s and early 90s. But nevertheless, those individuals are committed to lifelong therapy, which has lots of side effects. Even though they don't progress to AIDS anymore, they still have issues with this therapy. It would be much preferable to cure them of the virus and therefore rid them of the problem forever.
Thank you, Doctor. As a student, I've taken a few biology courses, and I think that I'm at least a year behind getting to where you are at this table. But you mentioned very briefly about the FDA and, you know, at least talking about the whole smallpox discovery. I'm just wondering, within your process, how do you feel that some of these regulations towards clinical trials, have they been helping you to safely move forward in this, or is it been kind of slowing down innovation that we could be doing in medical discovery?
Well, I don't view it as, you know, the last thing I want to do is come up with some sort of, you know, a vaccine that's going to hurt anybody. So, you know, the fact of the matter is that there are regulations there for a reason. You have to be very careful in making a human vector, not only the vector the vaccine that you're actually working on, but that you don't, in the manufacture of it, introduce any adventitious agents and so forth. You know, I think that we can make a safe vector, and I think we can make one that'll comply with all the FDA rules, and we have to convince them of that, and we're working hard. You know, one of the things that we're doing now in both the monkey model and to the extent in human in vitro work is trying to provide a database that will convince them that our vector is safe, and a manufacturing plan that they'll be convinced that it is safe too. So you know, they definitely are hurdles to get over, but on the other hand, they're important hurdles that make sure that when we go into humans that we're not going to do any harm.
Hello, Arnaud Simon, a City Club member and lifelong HIV activist. Have done some work in HIV funding advocacy with IAV before, so it's really fantastic to have good news like this showing up. As you know, also, as activists, we had to fight really hard against some of the pharmaceutical industry to start being able to lower the price of antiretroviral treatment to make them available to lower income countries, especially in sub-Saharan Africa. That has been possible, thankfully. That's how we have vastly increased access to treatment. I was wondering if you started to wonder about specific provisions in terms of patent protection, in terms of agreements with the pharma industry, to make sure that the price of this vaccine, when it actually comes on the market, will not be too high, so that it will not be actually accessible on the global scale. Thank you.
Yeah, absolutely. That's a concern for us as well. I mean, the biggest need is in sub-Saharan Africa, and in fact, you know, one of the issues is that there's not enough money, even with cheap drugs, to treat everybody in Africa. You know, a lot of our support has come from the Gates Foundation, who has a global access agreement that anybody who gets funding from the Gates Foundation has to sign. We have no problem signing that. It really is, the agreement is complicated, but it basically does what you suggest it does. It makes sure that the vaccine ultimately, under those agreements, that anybody who licenses the technology and goes to sell will have to comply with those agreements. It's made available in areas under the developing countries and areas that they're poor, that it's made available at the cheapest possible price.
Another question from the floor. Last week we heard from Dr. Baltimore that HIV is a, quote, wimpy virus. What about CMV?
Well, I heard that talk from Dr. Baltimore, and I think he was referring to the fact it's wimpy in the sense that it takes 100 sexual exposures or 200 to get exposed. I think Dr. Baltimore would agree that once you get that first infected cell and you get infected, it's not wimpy at all. So it's really it has vulnerabilities at the initial stage of infection, which is what we're shooting for, which is what his work is shooting for with this gene therapy for neutralizing antibodies. But that's really when once it gets ahead of steam, it's not wimpy at all. In fact, it's very robust. CMV is, all these viruses are different universe. CMV is also, it could be considered wimpy in that it doesn't cause disease, and except for handful of instances where 5 billion, 6 billion people are infected with it. But it has evolved to do what it does, and it's very successful at doing what it does. It's a sort of a stealth virus. It manages to infect almost all of humanity without causing much in the way of disease, which is really what a parasite, a highly adapted parasite, is supposed to do. And as I said before, this is a very highly adapted parasite. We've co-evolved with this particular virus. And so it's not wimpy at all, but on the other hand, we are understanding why HIV does what it does. We are understanding why CMV does what it does. It's a complicated virus. It's a much bigger virus, but we're able to manipulate it. And the safety, we're engineering safety into these vectors. Turns out that CMV can, even though it doesn't take very much to elicit the immune response, and we can make a virus that's completely unable to spread from one cell to another that still works as a vaccine. So, with those kinds of manipulations, we think we can make a vector that will be persistent, that will be safe, it will be unable to cause disease, but it will still have the desired immunogenicity or ability to elicit immune responses that we think will be protective.
Ted Kay, City Club member. You said that your research might help with the development of a new vaccine for tuberculosis as well.
Yes.
Does that happen in parallel with your work on HIV, or does it have to wait until you've solved HIV?
No, we're working on that now. It will be accelerated. I mean, we're developing a vaccine platform. We took on HIV from the beginning because the need was the greatest, and frankly because the funding was available for HIV to allow us to develop this platform. We are concomitantly taking this platform and assessing its ability to work in TB and in malaria. I don't have anything to announce on that yet, but that will be something that we'll have announcements probably in the next year or two on both those for both those pathogens. Assuming that if it does show efficacy similar to what we've seen in HIV, it would basically be the same platform, but with different, instead of HIV antigens or HIV inserts, it'll have tuberculosis or malaria inserts. So conceivably, this idea, which is different from all other vaccine ideas, might work for a number of diseases. In which case, this could be a, sort of a, one shop or one-stop shopping for some of the as yet unmet needs for vaccines against infectious diseases.
Hi, Liz Milan, and I was wondering, was there an age that you typically inoculated the primates, and was it more beneficial if it was a baby compared to an adult?
I'm sorry, I didn't quite hear that. Can you say it again?
Did you do different experiments associated with the age of the primate and inoculation?
The age of the primate. We tend to focus on most of our animals that we use tend to be young adults, but we do have evidence that it can work in older adults. We have not yet gone into very young, like the equivalent of babies yet with these vectors. But age doesn't seem to be an issue, at least in the range of what our target population would be. And by the way, the vaccine has been shown to protect against all ways you acquire HIV. It protects us through sexual transmission via the female genital tract between the rectal mucosa and even IV. So it doesn't seem to matter how you would be exposed to the virus, it still seems to work.
Another question from the floor. Is the HIV infection similar to typhoid fever in the fact that it takes thousands of units of virus to develop the disease, so that an individual with a very low viral load is not contagious?
Well, there's a whole area in the field of so-called treatment as prevention, and that is that individuals who have their viral load suppressed by drugs, and in some cases the few individuals who are able to control without drugs their viral load to that low, are going to be less contagious than ones who have uncontrolled infection. And so the idea is if you treat everybody, you'll have an effect epidemiologically on the disease. But none of that is foolproof. The only thing that's foolproof is to actually get the virus completely out of the body.
And maybe a provocative question here for you. In 1984, Secretary Heckler declared that vaccine for HIV would be ready for trials within 2 years.
Yeah.
Would you care to make a similar statement at this time?
No. I will say that we're targeting that the task that we've outlined in terms that we've given ourselves based on where we are now would take a minimum of 2 years to get into an initial clinical trial. Again, that's not an efficacy trial; that's a safety trial. It could easily be 3 years, and it depends. If the FDA decides they want X, Y, and Z, then we have to go back and do X, Y, and Z, and that could delay it. So no, I'm not going to make predictions. All I will do is say that this is our hoped-for timeline.
Well, with that, we've run out of broadcast time for further questions, and we'll have to stop for today. Please join us next week for the results of the Oregon Values and Beliefs Survey. And as we close today, please join me in offering our sincere thanks to Charles Wilhoite and Dr. Picker. We are adjourned.